Hi everyone, today we're doing phospholipids. I'm really sad because I recorded an entire video about phospholipids and it just couldn't get saved. Anyways, what comes to your mind when you first hear the word phospholipids? I'm sure it is cell membrane because phospholipids they form a great chunk of your cell membrane. Um, Now phospholipids, when we were doing the classification, I clearly told you that phospholipids they fall under the category of compound or complex lipids, right? Compound or complex lipid, which was the second category of lipids that basically comprises of, or that's where your phospholipids fall under. Now, phospholipids are further divided into glycerophospholipid and sphingophospholipid. When you talk about sphingophospholipid and glycerophospholipid, so one thing that distinguishes the two is the alcohol used. So in glycerophospholipid, you have glycerol as the alcohol, and in sphingophospholipid, you have sphingosine as the alcohol. When you talk about the structure of phospholipids, so first of all, you have fatty acid, then you have your alcohol, then you have, very important, you have your phosphoric acid, or you can say phosphate group, and then you have the nitrogenous base, right? So I told you already that glycerophospholipid and sphingophospholipid they differ in the alcohol used. So glycerol is in glycerophospholipid and sphingophospholipid has sphingosine. Let's just quickly get done with sphingophospholipid because it's a very small category and its example is sphingomyelin. Now, as the name suggests, it helps with the myelination. This is the role of sphingomyelin. Now, moving back to glycerophospholipid. You have a lot of variety of glycerophospholipids. First of all, you have phosphatidylcholine, also known as lecithin. Then you have phosphatidyl ethanolamine, also known as cephalin. Then you have phosphatidyl inositol. Then you have your phosphatidyl serine. You also have phosphatidyl threonine. These are the common ones and easy to remember. But there are three more that we often tend to forget. One is cardiolipin, then you have plasmalogens, and you also have platelet activating factors. These are often neglected even though they're super important. Now, when you talk about lecithin, I already told you it has a choline nitrogenous base, so they differ in their nitrogenous bases. This thing, right? Nitrogenous base. If it's choline, then that means we're dealing with lecithin, and if it's ethanol amine, that means we're dealing with our cephalin, right? And lecithin and cephalin, lecithin it forms a very important part of your surfactants. Yes. Now again, phospholipids. Coming back to phospholipids as a huge, as a major category, glycerophospholipids and sphingophospholipids. I'm referring to both of them right now. Phospholipids, they are amphipathic in nature. Why do I say that they're amphipathic in nature? Because this means they have hydrophilic as well as hydrophobic part. Their hydrophobic part being the fatty acid chain, and hydrophilic is the polar head that comprises of glycerol, and it also has nitrogenous base in it, right? And phosphoric acid. So if you can recall, this is the phospholipid structure. So it has phosphate or phosphoric acid, glycerol, and a polar group of nitrogenous base. And this is the hydrophobic fatty acid tails. And this is how they form the cell membrane, the bilayer phospholipid bilayer, right? The bilayer membrane. Now, um, also, now phospholipids, they do not just have the role of cell membrane, but they also serve as a reservoir for intracellular messengers. Other than that, they help to anchor different sort of proteins to the cell membrane. They also make up the lung surfactants, and they also are essential component of the bile, where their detergent-like properties helps with the cholesterol solubilization. Now, when you talk about lecithin and cephalin, so here's their function. Lecithin helps form and stabilize cell membrane and acts as an emulsifier for fat digestion. Cephalin, on the other hand, it supports cell membrane structure, yes, and it plays a role in the blood clotting. And if you are asked to distinguish between, okay, we'll also come to cardiolipin and all of that. Actually, let's just move to cardiolipin and plasmalogens because it's not important for you to know the function of all of these individually. Just saying that they form they form an important chunk of your cell membrane. Just saying that would be enough because they help with the structure and function of the cell membrane, the stability of the cell membrane. Well, moving on to cardiolipin. What exactly is cardiolipin? Cardiolipin is basically when you have phosphatidic acid here and you have a phosphatidic acid here, and in the middle it's held together by glycerol. So basically there's an ester bond that's keeping the two together. So phosphatidic acid, there are two phosphatidic acid, and then there's a glycerol in the middle. This is your cardiolipin structure. So it's very important for you to know it. Now, you may be wondering what exactly phosphatidic acid is. If you do not know, so these three collectively are known as phosphatidic acid. Right. Now, a cardiolipin is basically two molecules of phosphatidic acid esterified to their phosphate groups to an additional molecule of glycerol to form cardiolipin. The cardiolipin, where is it found? It's normally found in the membranes and especially and particularly the membrane of the inner mitochondrial membrane. So the inner mitochondrial membrane, and what do you find in the inner mitochondrial membrane? We have electron transport chain. So cardiolipin, it has a major role in maintaining the structure and function of the respiratory complexes which are a part of the electron transport chain. So this is a very important feature of cardiolipin. What does it do? It maintains a structure and functional aspect of the respiratory complex of the electron transport chain. Now, cardiolipin, there's another very important thing about it in regards to its clinical correlation, that this has antigenic property. What again? Cardiolipin, it has antigenic property. Now, what do we mean by that? I'll give you an example of a clinical condition which is syphilis. What happens in syphilis? Basically, painless genital ulcers develop in this disease, and it's a sexually transmitted infection. Now, in this, what happens is that there is a bacteria called T. pallidum, in which what happens is that once this bacteria enters the body, it starts breaking down or attacking on the tissues. And what happens is that the cellular mitochondria, it's damaged and it breaks apart, and the cardiolipin, it is released into the blood. As long as cardiolipin is protected and safe and hidden in the inner mitochondrial membrane, our body doesn't detect it. But obviously once it starts flowing freely in the bloodstream, our body's immune system gets activated and it identifies this as a foreign agent, right? And for an extra information, this bacteria, T. pallidum, it also has cardiolipin as an antigen on its surface. So when the body sees that, oh my God, what is this foreign agent? It starts working against it. It responds towards cardiolipin with anti-cardiolipin antibodies. So this is very important for you to know that body creates anti-cardiolipin antibodies against this and it starts attacking the cardiolipin. So this is the clinical correlate with cardiolipin. Now moving on, plasmalogens. Whenever you think about plasmalogens, so if you can recall, we studied about phosphatidyl. First of all, I'll tell you the structure. So phosphatidyl ethanol amine, which is cephalin, right? Now phosphatidyl ethanol amine. Um, Basically, plasmalogens have exactly the same structure, and the only difference is that at carbon 1, you need to have a double bond plus obviously ether linkage. Yes, so if you can recall from this classification that I already explained to you that plasmalogens, they are phosphatidic acid plus ethanolamine. So basically, it's the lecithin, right? Sorry, choline, right? And then you have your carbon 1, which is unsaturated fatty acid, and there's ether linkage, which is attaching it. So what is plasmalogen? It is basically your phosphatidyl ethanolamine, which is cephalin. Phosphatidyl ethanolamine, which is your cephalin. Now moving forward, yes, here. So it is basically your cephalin, which is um, at the carbon one, you have a double bond and there is an ether linkage. This is your plasmalogen structure. Now, what is its function? This basically ensures that the cell membrane is protected from all, by all means, it's protected, and it is flexible. So phosphorylethanolamine is a protective, flexible lipid membrane that helps the brain, heart, and nerves, very important, to work properly, and it also acts as an antioxidant. Now, what does this mean? For example, if there's any dangerous radical that is there in our blood and that's attacking the membranes of these three structures, so what does our plasmalogens do? It basically works against them and protects them because antioxidant, it has antioxidant properties. Now moving on, this is plasmalogen. So it has, when the fatty acid at the carbon one of glycerophospholipid is replaced by unsaturated alkyl group attached by an ether rather than an ester linkage to the core glycerol molecule, an ether phosphoglyceride known as plasmalogen is produced. Also, if you can recall from the synthesis, so it is given the name of alkynyl because the first ether linkage at first carbon, which is having double bond, that's known as alkynyl. So this is the reason it is one alkynyl. Yeah, so plasmalogens, what do they do? Protective, flexible cell membrane with antioxidant properties. Moving on to the plasma activating factors. Now, when you talk about plasma activating factors, you should know that what do they do? These basically also structure-wise, they are ether glycerophospholipids. And what's the function? First of all, they all, clump together and help with the blood clotting. So they all clump together, and then the second thing they do is they cause or they trigger inflammation. And after that, what they do is they make the blood vessels very leaky, and after that, they direct the white blood cells to the site of infection. So this is their very important role. Now, again, it's very easy for us to remember the remaining ones, but usually we tend to neglect this, even though they're so important. I already told you cardiolipin, it has antigenic properties, and then cardiolipin, this is its structure, and it maintains the structural and functional integrity of the electron transport chain, which is present in the inner mitochondria. How about plasmalogens? They ensure that a protective environment is given to the cell membranes, particularly of our brain, of our nerves, and of our heart, and also they ensure that they're flexible. And plasmalogens, they have antioxidant property to themselves. And platelet activating factors, they trigger inflammation, they cause blood clots, then trigger inflammation, then they make the blood vessels leak, and also they direct the white blood cells to the site of infection. So we often tend to neglect this, even though they're super important. And sphingomyelin, they help with the myelin sheath, right? And choline and cephalin, for now, you need to just know that they basically help to stabilize the cell membrane. And for lecithin, you can remember it's emulsifier for fire digestion. And remember, lecithin has choline as the nitrogenous base, and cephalin has ethanolamine as the nitrogenous base. And cephalin, it helps with the blood clotting as well. So, coming back to the main question, what do you think of when you first hear the word phospholipids? So we started with cell membrane. Now it's pretty clear to us that it's not just cell membrane stability, that's not just their sole function, but they also help with so many other things if you individually go and discuss them. And yeah, this is what I had done before. Sadly, my video it got deleted. But anyways, yes, so that's pretty much it.